Okay, everyone. We'll start on the second part of chapter 16 with inflammation. So we'll go through this example of inflammation. Basically, inflammation is the body's response to an infection. And so in this example, we can see that your intact skin has been damaged and with a cut, and the cut has allowed bacteria to get beneath the surface of the skin. What initially happens with inflammation is that those damaged cells would release histamine, and that would cause the capillaries to dilate, okay, or get bigger. That would be vasodilation. And then you have blood at the surface of the skin, which would cause redness and heat. Also, when the capillaries dilate like that, a capillary is just basically a single cell thick, and because that is, fits the function of the capillary, right, which is diffusion of gases. So you have oxygen diffusing from the red blood cells inside the capillary through this just one cell layer thick capillary wall, and the cells are connected together, the cells of the capillary, by these junctions, which become leaky. And so fluids can leak out of the capillary, and that would be what would cause the swelling or the edema. Phagocytes can also adhere to the capillary wall and then actually move through the capillary wall, okay, through these same junctions. And they are going to move through the capillary wall, and they would actually chemotax, okay, move toward the chemotactic chemicals that are being released by the microbes. And then you would have the whole process of phagocytosis, which we just talked about, occurring, okay, where they would adhere, ingest, digest, etc., okay, after the chemotaxis. Okay, so the functions of inflammation are to kill the microbes, okay, and the phagocytes would do that. The phagocytes would also clear the debris, okay, and then you would also have repair of the tissue, and this can occur very quickly. Because that repair, you also want to wall off the infection so that it doesn't become more invasive and get into deeper tissue. And so the repair would start at the beginning of the inflammatory process, but it wouldn't be able to finish until the very end, until the infection was cleared. But, all right, since you do have that rapid tissue repair, you can actually develop scar tissue. Okay, and scar tissue usually is not functional tissue, and it could actually impair the function of the tissue once the damage is repaired. Okay, so histamine would be released from the damaged cells and cause the capillaries to vasodilate. Okay, and that's where you would get the edema. Okay, where you've got like the leaky fluids coming from the capillary. You'd also get redness. Okay, and heat. The phagocytes are going to move through the capillary wall and chemotax toward the microbes, okay, which are releasing these chemotactic chemicals. And you would have the whole process of phagocytosis occurring, okay, in inflammation. If you have an abscess form, all right, so it would be like the skin was repaired before the infection was completely cleared. The pus that accumulates on the inside of the abscess is actually dead white blood cells that were fighting the infection. So it's better to like keep a wound open so that it can repair from the inside out so that you don't have an abscess forming. Okay, and then repair and regeneration. You want to repair the destroyed tissue and also wall off the infection so that it doesn't become more invasive or go into deeper tissue. Fever is actually pretty interesting. Fever is more of a systemic response. And so it actually, the resetting of your body temperature occurs in your brain, specifically the hypothalamus. So fever is defined as a oral temperature that's greater than 100.5 degrees Fahrenheit. Okay, we're talking about Fahrenheit right now. And in response to pyrogens, okay, exogenous pyrogens, it's kind of old school terminology, but these would be like exotoxins and endotoxins from bacteria. Okay, so just to review, hopefully you remember what an endotoxin is, right? We talked about like lipid A being an endotoxin. So endo is part of the bacterium. And then an exotoxin would be something that the bacterium produces. Okay, so oftentimes exotoxins are enzymes. So in response to those types of things, your macrophages would actually release cytokines, which would cause the neurons in your brain to release prostaglandins, and then the prostaglandins would reset your body's temperature. The hypothalamus would reset your body's temperature to a higher temperature. Okay, and then you, in response, would feel cold, and you would feel chilled because your body has been reset to this higher temperature, and you're not there yet. So you would start shivering, and your body temperature would rise. Okay, and then when your fever breaks, okay, and your body temperature returns to normal, but you're warmer than that, you would actually be sweating, right? Okay, so you would be sweating when your fever breaks. Okay, so this is your body's response to a systemic response to an infection. I actually included the slide right here about the benefits of fever. Okay, now let me just clarify that you shouldn't just let a fever rage, right? But you should probably definitely take some, some type of medication, right? But there are some benefits to having like a low grade fever. And the last thing that I mentioned there is that if you feel bad, right, you'll rest and you'll allow your body to fight the infection. But there are actually some other benefits, and one would be that the elevated temperature. Okay, think about like the optimal temperature, right? Remember that bell-shaped curve, the optimal temperature for cell growth or for enzymes to work. If the optimal temperature for a microbe that grows in our body would be like our body temperature, which would be 37 degrees. But, okay, if you have a fever, say it's 39 or 41, that is actually not the optimal temperature for the growth of a pathogen. Okay, it's not the optimal temperature for microbial enzymes or their toxins, right? We just mentioned that the exotoxins could be enzymes. And so, okay, at that higher temperature, you're not going to have the bacterial growth would slow, and also the toxins or the enzymes might not be functional. Okay, they could be inactivated. Now, fever or temperature, right, is a catalyst. And so temperature, an increase in temperature could actually increase the rate of some of our chemical reactions. Okay, so it could actually enhance our immune response. Phagocytosis is enhanced. The production of interferon, okay, which we haven't talked about yet, but interferon is an antiviral. Okay, it's increased with a temperature. And also the breakdown of lysosomes. Okay, remember lysosomes are kind of like this recycling type of organelle within our cells, which have a lot of hydrolytic enzymes in them. So lysosomes would be broken down inside of cells, and that would kill those infected cells in a person who had, some type of infection and was having a fever. Okay, so there are some benefits to fever, all right, but okay, in general, you wouldn't want to let your fever just, rage for a long period of time. Okay, that could actually be damaging. Okay, so here's interferon. Okay, so interferon is a molecular defense. Interferon is a protein. And when you have a virus infecting a cell, that virus would initiate a signal, okay, like a signal transduction pathway to, okay, the nucleus of the cell to express a gene, okay, and the gene is the interferon gene. So interferon would be synthesized by that original infected cell, okay, which by the way will die. Okay, that original infected cell will die, but it makes the interferon protein, which then diffuses to neighboring cells and stimulates a new signal transduction pathway for those neighboring cells to make an antiviral protein, okay, which is going to fight the viral infection. Okay, so in this example right here, binding of the interferon stimulates the production of the antiviral proteins, the AVPs, and then, okay, in the third cell of this slide, when the virus infects the cell, okay, that has produced these AVPs, the AVPs inactivate the viral DNA, okay, or in this case RNA, right? The viral mRNA is digested by the AVPs, okay, the antiviral proteins. Okay, so complement is another molecular defense. Complement actually is about more like a set of 30 large regulatory proteins that are made by your liver, and they circulate in your blood unactivated. Okay, we'll see that complement works as a cascade. So when one complement protein activates the next complement protein in the pathway, you have an amplification of the effect. Okay, and so what that means is that, like, when the first complement protein is activated, it will activate 5 of the next complement protein in the pathway. And then each one of those 5 complement proteins is going to activate 5 of the next complement protein in the pathway. So you go from 1 to 5 to 25 complement proteins being activated. That's the function of a cascade. It's a set of reactions in which the effect is amplified with each step. Okay, we're going to see that complement will enhance phagocytosis, all right, by opsonization. It will lyse microbes. Okay, there's a membrane attack complex that will lyse microbes, and it generates peptide fragments. Okay, that's what complement does. It activates the next complement protein in the pathway by lysing it. Okay, breaking it into 2 peptide fragments. And those peptide fragments can regulate inflammation and other immune responses. Okay, so there are 2 pathways. There is the classical pathway, which is initiated by antibody binding to antigen. And then there is the alternative pathway. Okay, so in the alternative pathway, there are no antibodies. Okay, there are actually different complement proteins that bind to the cells and activate a third complement protein in the pathway C3. And so with the alternative pathway, you're not using all of the complement proteins that you would be using in the classical pathway. In the alternative pathway, we'll see that we get to a certain point in the pathway of the classical pathway, which is similar to the alternative pathway from there on out. Okay, so the beginnings of these 2 pathways are different, but once you get to a certain point, they're both the same. Okay, so I know, like, your heads are probably spinning, like, how can I memorize all of this? Like, it's C1, C4, C2. Actually, these proteins were named in the order of their discovery, not in the order of the way they function in this pathway, so they're out of sequence. But it's actually only C4 that is out of sequence, okay? And then from there on out, it's like, 2, 3, 5, right? We skip 4, 6, 7, 8, 9. Okay, so it's not that hard to memorize. You are going to need to memorize some of this information because there's no way I can test you on this without you having memorized it. So, like, for example, you would need to memorize that C3A is going to enhance inflammation, okay? And I'll show you that in just a minute. And that C3B is an opsonin, okay? And I'll show you that. We talked about opsonization, right, of an antiphagocytic capsule, okay? We'll look at opsonization in just a second. We'll look at that again. C3b is also going to initiate the rest of the pathway, okay, which is going to lead to cytolysis. Okay, we talked about that, right? That's when the cytoplasm is lysed, right, and the cytoplasmic contents leak out, and you have a bursting of the cell. Okay, so C3b splits C5, okay, which would activate it, okay, and then C5b would bind to C6, 7, 8, and a bunch of 9, C9, to form a membrane attack complex or a MAC complex. Okay, and this is also referred to as a C9 complement lesion because basically it's a hole in the cytoplasm, or I'm sorry, the plasma membrane of the cell, which causes the cytoplasm to leak out, extracellular fluid to rush in, and the cell to rupture, okay, or lyse. Okay, so we'll also see on this figure that C3A and C5A play a role in inflammation because they bind to mast cells and cause them to release histamine. Okay, we just talked about histamine's role in inflammation. And then C5A can also establish a chemotactic gradient for phagocytes to travel toward. Okay, so we'll see that on our figure that's coming up. Okay, so this is the classical pathway, right? So you have antibody, okay, binding to antigen, okay, which is how the classical pathway is initiated. It would bind to C1, okay, and activate it, okay, and then C1 is going to split C4, okay, and activate it. C1 also splits C2 and activates it. And then C4b and C2a together would activate C3. Okay, C3b is an opsonin. And so look at this phagocyte at the bottom of this figure. There's a bacterium that has those Y-shaped antibodies bound to it, right? But then it also has the little kind of yellow disc looking C3b bound to it. Okay, so this is what an opsonin does. It would coat a bacterium, and then it makes it easier for the phagocyte to recognize that bacterium because it's not really recognizing the bacterium. The phagocyte is binding to the complement protein, or it's binding to the antibody. Okay, so it's not binding directly to the bacterium, okay, which might have an antiphagocytic capsule that it doesn't recognize, but it definitely would recognize, that stem part of the Y-shaped antibody. It could recognize that easily, or it could recognize the C3b. Okay, so the phagocytes actually have receptors for antibodies, and they have C3b receptors. Okay, so if a bacterium is opsonized, is coated with those proteins, okay, which are called opsonins, right? So C3b would be an opsonin. An antibody could be considered an opsonin. Okay, then you have this process of opsonization, which is going to enhance phagocytosis. Okay, C3b is also going to initiate this MAC complex, membrane attack complex. Okay, so we'll talk about that in 1 second. But C3a and C5a bind to mast cells and release histamine, okay, which leads to vascular permeability, okay, vasodilation in inflammation, right? Okay, and you can see how the fluid from the capillary is leaking out, Okay, that would be what would be causing like edema, swelling. And then C5a also sets up a chemotactic gradient for phagocytes to travel toward. All right, and there you can see a white blood cell moving through the capillary wall, okay, to go to the site of infection. Okay, so getting back to the third function of complement, which would be this membrane attack complex, okay, which would cause lysis of the cells. C3b is splitting C5 into two parts, okay, C5a and C5b, okay, and that activates it. And then C5b binds to C6, C7, C8, and a bunch of C9 to create these membrane attack complexes or C9 complement lesions. Okay, and that would cause cytolysis. Okay, I'm just gonna go ahead for the alternative pathway. Okay, it's gonna be exactly the same once you get to C3, but instead of using C1, C4, and C2 and being activated by antibody binding to antigen, the factors B, D, and P actually bind directly to lipid A or polysaccharides, and they, okay, bound to the surface of the microbe, would then activate C3. Okay, and then from there on out, all right, it's exactly like what we just talked about, okay, with inflammation and opsonization and the membrane attack complex. Okay, I just wanted to talk about some other proteins. These are iron binding proteins. The first three on this list, transferrin, lactoferrin, and hemoglobin, those are our iron binding proteins. Okay, so transferrin binds to, it's a protein, okay, that's in blood plasma that binds to iron. Lactoferrin is another protein. Okay, so these are all iron binding proteins that's present in saliva and mucus and milk, okay, that binds to iron. Okay, and then of course hemoglobin, which is packed into our red blood cells, is what is bound to iron, and that's what carries oxygen. That fourth protein, okay, the siderophores, those are proteins that pathogens, okay, that microbes have, that actually have a higher affinity for iron than our own iron binding proteins. So they can strip iron away from our iron binding proteins. Okay, so usually I would ask, what would a microbe need iron for, right? And then, hopefully a student would say that, iron could possibly be used as a cofactor for enzymes, that there are iron proteins, iron sulfur proteins in the electron transport chain. Okay, the cytochromes are iron proteins, right? That can exist in the oxidized and reduced form. So microbes need iron too, and these proteins actually can take iron away from our iron binding proteins because they have a higher affinity for the iron. In lab, okay, can't remember if we've already done this, but we're going to be looking at beta hemolysis. Beta hemolysis is when you have the complete lysis of red blood cells. And S. pyogenes, Streptococcus pyogenes, is beta hemolytic. Okay, so it completely lyses the red blood cells. So it has these proteins called hemolysins. Okay, so lysing the red blood cells, right? And that is used to obtain iron from the red blood cells. Okay, from the red blood cells, which are packed with hemoglobin, which have a lot of iron. Okay, so this would be another mechanism for obtaining iron from our iron binding proteins. Okay, but we do have our defenses against pathogens, and these are actually kind of newly discovered molecular defenses of our innate immune system, these antimicrobial peptides, okay, or AMPs. So a peptide is a short protein, like I would say less than 50 amino acids long. Okay, these antimicrobial peptides can be between 12 and 50 amino acids long. They have a broad spectrum of antimicrobial activities. They also work together synergistically. Okay, and so, what I mean by that is like 1 + 1 = 5, right? There's synergy there. Okay, so they work better together than they do individually. We actually produce these antimicrobial peptides when our toll-like receptors bind to, remember the toll-like receptors? Okay, when they bind to the pathogen-associated molecular patterns in microbes, the PAMPs, our cells will produce these antimicrobial peptides. Okay, so you can see how these different parts of our innate immunity work together. Defensins are actually antimicrobial peptides. They are present in our mucus and other fluids, and they would kill microbes by creating pores, okay, which would cause cytolysis. These antimicrobial peptides, they are actually even making I guess you could call them antibiotics, which are antimicrobial peptide mimics. They're synthesizing mimics of these antimicrobial peptides as antimicrobials. Okay, and